So thank you very much, Maura, for those very kind introductions. And thank you, everybody, for attending this, I have to say, really impressive webinar. This is, as we all agree, a very important topic. And I have to thank all the organizers and the board of ICE-HBV for putting Delta on the agenda. I think it's so important to talk about this disease, not only to discover the disease, but we have to talk about treatment. And as treatments are available, we have to identify the patients. We have to share experiences, how to manage these patients. And therefore, I think these webinars are extremely important. So I've been asked to discuss this evolving landscape. I'm sitting right now here at Hanover Medical School in my office. You see, we are a quite green city, and we have quite some groups in Hanover working or treating patients with Delta. This is the infectious disease ward. These are the outpatient clinic, but also doing some translational work to understand the pathogenesis. These are my potential conflicts of interest, which includes companies developing drugs against HBV infection. And I have to acknowledge many people who are working here at Hanover on Delta, but also our collaboration partners in the German Center for Infection Research, including Maura, in different consortia, including the European-funded DSOLVE consortium. So we have, when we talk about treatment, I've often asked about what are the options. I can refer to this guideline and stop my talk because last year we released Mauricia Brunetto. She coordinated the EASL clinical practice guidelines. Pietro was one of the reviewers of these guidelines. And you can see here this main figure. It states that we have two options. We have pegylated interferon alpha. We have Bulevirtide. We may combine this, and the more advanced the disease, this figure suggests to use Bulevirtide, and in the patients with a milder disease, you may use pegylated interferon. This is basically what's written at that time without too much evidence, I have to say, and we may discuss this later also critically, whether this is really holds true in the year 2024. But I think it's a nice scheme, and it's for the very first time that we really have international guidelines. My message today is really that, and that in contrast to hepatitis C, where we have simplified treatment, one-size-fits-all strategy, this is not the case for Delta, and for various reasons. Access to treatment, but also if treatments are available, one treatment may not be the best choice for every treatment. So we need personalized concepts on all levels. Elisabetta mentioned diagnostics as but also surveillance. Let's say to screen everybody may not be the ideal approach in each country. We really have to individualize here the concepts. And when you just look at the natural history, this is where we compared B versus Delta. The question is really, which are the factors determining if a patient develops HCC, decompensation, or death? Because some patients may have a rather mild disease. Not every patient is developing endpoints. And therefore, we need tools to stage the disease, to predict patients with a higher likelihood of disease progression. And I have to highlight two publications which I think are really important for clinical practice. The first one is that Lisa Sandman and colleagues from different centers in Europe really showed that liver elastography is a very useful tool, a robust tool to stage fibrosis. And these have been the cutoffs that have been established in that paper. So to decide, does a patient need urgent treatment, yes or no, elastography is sufficient. It's a robust method. The second point is obviously which methods really predict clinical endpoints. And I have referred here to a paper by Matthias Jachs and colleagues from Vienna and Hanover, who really show that non-invasive tests predict endpoint developments in advanced HDV cirrhosis, including portal pressure gradient measurements, a very nice paper. But this is important if you want to personalize treatment approaches. And just to add one point to Elisabetta's very, very important comments on issues on HDV RNA testing, one answer to her issues were, well, we still have a WHO standard. You may just calibrate all your assays with extraction methods, etc., to the WHO standard, and then all problems are gone. That should be or you have a conversion factor or something like this. And I want to highlight one point, that Delta is really variable. And even if you use different assays, these are published here two different RoboGen assays, 2.0, 3.0, and with different extraction methods. You can see here, if you apply these four tools or combinations with the WHO standard in dilution, this works quite fine. And for some clinical samples, it also works quite fine. But in other clinical samples, these were random routine clinical samples, you have suddenly huge variabilities, more than 2 logs in some cases. And this really shows a problem. There will not be a conversion factor related to the WHO standard to overcome this problem. This is why do I spend time on this? Because when we talk about treatments, don't switch between assays. You cannot predict whether this patient or this patient or this patient is, or in which group this patient is. We have to stick with one assay, and there will not be, like in hepatitis B, when we start treatment, let's say with a cutoff of 2,000 international units, then you have to think about treatment. We will never, ever have one HDV RNA cutoff 1,000, 10,000 to decide to start treatment, yes or no. This will never work in Delta. We have the final point we have to consider, that Delta is really heterogeneous. We all know that Delta is heterogeneous. We have the different genotypes in Africa. The point is that even the point when to decide antiviral treatment, this may differ in different regions. Annika Wranke just published Delta from the International Network registry, where she could show that the outcome, the likelihood to develop an event over time is different if you look into different regions, Europe versus Africa, versus Pakistan, versus South America. So this is another point we have to, that there is not one size fits all treatment. We have to be flexible. Which treatments do we have? We should not forget that these patients are co-infected with B, so that we should apply HBV treatment approaches. At this stage, we have interferon, we have Bulevirtide, we can combine them, and then we can evaluate alternative treatments in clinical trials, and we'll mention some of them in the end. Interferon. I think what has to be highlighted, we have a huge experience with interferon, and it's a well-established treatment options. And it should be used when Bulevirtide is not available. We'll spend a lot of time talking about Bulevirtide, but there may be people watching this webinar where simply they do not have access to Bulevirtide. And therefore, guys, don't forget interferon. We know some host and viral factors which may predict response. We even have weak data on some kind of response-guided therapy, which we have been using in hepatitis C treatment quite some years ago. But it's a drug where we should remind ourselves what has been generated in evidence. We know that roughly 25% of patients may achieve HDV RNA suppression. We know that there is relapse after the end of treatment. We know that there may be even late relapses after several years. This has been shown by several sites. So if you treat a patient with interferon, follow your patient. But the most important point is, if you treat your patient successfully with interferon, you're doing a good thing, because there have been several cohorts, Pietro and the Milan team published this 15 years ago, and other people later, that if you are HDV RNA negative after or at the end of treatment, and even if you have a late relapse, you still have a good response, while if you do not achieve a response, you have a higher likelihood to develop clinical complications. And there are factors which may guide your decision points. Lisa Sandman summarized this. Cirrhosis is not a negative factor for interferon response, and so on. I refer to this paper if you have to use interferon. But now let's talk about Bulevirtide. You all know that Bulevirtide receives conditional approval by EMA in summer 2020, full approval in 2023. It's available, meanwhile, in several European countries. There are differences in reimbursement practices. So it's in some countries only, let's say, kind of compassionate use programs, and others you have to ask the insurances, and others you can just prescribe it without asking anybody's. It's a quite, let's say, diverse pattern of approval status, which changes every week in Europe. To remind those guys who are not working on a daily basis with Bulevirtide, it's a peptide targeting the pre-S1 region, which is a binding site for HBV, HDV receptor, which is NTCP. It specifically binds NTCP, and therefore It's also very safe because it's only targeting hepatocytes. It shows a strong inhibitory potential, and again, it exclusively targets liver parenchymal cells. It's a peptide which needs to be injected once daily. This is important. We have data from the phase 3 registration trial, a trial that compared 2 versus 10 milligram of Bulevirtide daily injections. There was one referred treatment arm. The primary endpoint was 48 weeks of treatment, and the primary endpoint was a combined response, virological 2-log decline with ALT normalization, which were achieved at that time in 45% and 48% of patients respectively, and this basically was the basis for also full approval by the European Medical Agency. The 96 weeks data have just been published in the Journal of Hepatology, where you could see that with continuous treatment, the number of patients being virological responder increased, and after 2 years of treatment, only few patients, less than 20% overall, did not achieve at least a 2-log HBV RNA decline. You can also see that ALT normalization was achieved in almost 2/3 of the patients after 2 years of treatment with Bulevirtide after 2 years. It's important to highlight that this treatment is associated with improvement in clinical markers. Liver stiffness improved. Pietro and his team and Elisabetta showed already 2 years ago that markers of portal hypertension improved in 18 patients with significant portal hypertension, which I think is very nice. Elisabetta then presented at EASL a very large cohort of 167 patients recruited at 37 centers, basically confirming response rates that were observed in the phase 3 clinical trials. And these were all patients with compensated cirrhosis. You can see here these respective numbers with virological undetectable rate, virological response, biochemical response. Remember, I told you that 64% had normal liver enzymes after 96 weeks in 301, and Elisabetta presented 63%. And so the numbers are quite remarkable, even in patients in compensated cirrhosis. What is then important that there is not always 100% link between virological response and biochemical response. In our German experience, we still had patients, even at patients who did not achieve a complete virological response, we had remarkable improvements in liver enzymes. That is important, and when you have a closer look into the clinical registration trials, and Pietro presented an analysis where different HBV RNA cutoffs were evaluated, and then they look into the biochemical response rate. So if you have a patient who has undetectable HBV RNA, you can see here that 86% of patients had ALT below 1.5 upper the limit of normal. But even a patient who had only below 10,000 units of HBV RNA, you still have 83% achieving this biochemical response. So there is no strict correlation. And for us in clinical practice, guiding treatment on Bulevirtide treatment, for me, actually, ALT is more important than HBV RNA, which is also some question mark we may discuss in the panel later on. The other point that Elisabetta demonstrated at EASL is really important. She tried to match her patients with a historical control group. And this analysis suggested that there may be a benefit in terms of clinical complications by Bulevirtide treatment in the long-term outcome. This does not look too dramatically, but if you look in this table, you can really see that the number of patients achieving who started off with Child-Pugh A5, who had then after 2 years hepatic decompensation. There was not a single patient in the Bulevirtide treatment arm or group that achieved this clinical complication, while this occurred in the untreated cohort. So there seems to be a rather early effect already on decompensation. This was not seen for HCC, and Elisabetta may comment on details on this important study. What is also for clinical practice important. What is happening if a patient with decompensated hepatitis D, which I think is also very important. And Christopher Dies-Fricke summarized from different centers data here in this group, and he could show that if a patient started off with Child-Pugh B stage, that half of them improved the MEL score over time, which I think is clinically highly relevant. And to me, even though Bulevirtide is officially approved only for patients with compensated liver disease, ethically it may be difficult to withhold this treatment from patients with decompensated cirrhosis. Bulevirtide and interferon, there may be synergistic effects on HDV RNA. The advantage of this combination may be that we have also some effects on HBs antigen, while Bulevirtide monotherapy does not induce any S antigen decline. The data have been presented by Tarek Asalat last year at ASLD of this combination trial, which compared early combination with a tail of Bulevirtide monotherapy. The primary endpoint was what week 100 off treatment response, and here these are the basically HDV RNA undetectable rates. You can see here that this was at this stage quite remarkable, and this suggests also that the 10 milligram group might be better than the 2 milligram group. The final data have been published in the New England Journal of Medicine released during EASL and refer to this paper, as I don't have time to discuss details here now. I think it's quite important and impressive data that for a subgroup of patients, combination may be beneficial, but keep in mind that we only have 2 milligram available at this stage. We cannot prescribe 10 milligram. The question then arises, can Bulevirtide monotherapy be stopped? Pietro and his group published this very important case where Maura Dandri contributed with this very nice intrahepatic stainings, suggesting that indeed with continued monotherapy, then if you put a needle in this patient, delta was cured, even though S antigen was still present. So is this only a single patient? Does this occur more often? From the 204 study, we have now additional information, because there was this group with monotherapy with 10 milligram who were then also stopped after 96 weeks. And you can see here at the end of treatment after 96 weeks, 22% of the patients had undetectable HDV RNA, and indeed some patients maintained this after in the long-term follow-up. There were relapses, yes, but also in the other groups where you could see relapses. So in single patients, an off-treatment response can be maintained. Just some final words on the drugs in development. Interferon lambda, unfortunately, had to be stopped because of flares, so we don't have data on lambda. We have data seen on siRNAs against HBs antigen. If you use this alone, it works in principle. S antigen declines and also HDV RNA declines, but we have seen in patients with high HBs antigen levels flares in ALT, as you can see here. These were no beneficial flares in the long term, but when you stop treatment, you can see here that these flares disappeared. This is something which needs to be taken into account. But if you have patients with low S antigen levels as baseline, long-term treatment may be an interesting option, which we presented this year at EASL. Then there have been data on siRNAs against HBs antigen in combination with anti-HBs monoclonal antibodies. The VIR data have been presented by Tarek Asalat during EASL. Also quite interesting response. You can see here the combination group of the siRNA with the monoclonal, quite impressive HDV RNA declines, also ALT declines. You can see here the number of patients being undetectable or having very low levels of HDV RNA at week 24 is really impressive, I have to say. A very interesting treatment opportunity. The interesting point is that also other monoclonals against S antigen have been tested in Delta. We've seen here the BJT778 data at EASL at a late-breaking poster. Also here, very impressive HDV RNA declines in these patients being treated with 300 milligram of this antibody. And we have seen the year before an anti-PS1 monoclonal antibody by this Chinese company, also showing quite impressive HDV RNA declines. So the concept that a monoclonal may reduce S antigen and HDV RNA has now been shown for 3 different antibodies. We have seen nucleic acid polymer data quite some years ago in a paper, the first 11 patients published in Lancet GH 2017. We have seen several case reports presented during recent meetings, no full paper yet. Interesting approach, which we may discuss during the discussion. And this is basically my summary of the ongoing clinical trials. Lonafinib, where the phase 3 trial has been completed, but no further trials are currently planned. To my knowledge, Lambda, I mentioned the phase 3 trial had to be stopped. Nucleic acid polymers, we have seen additional case series during recent liver meetings and have to see how this will be further developed. For the other approaches, the SI and the strategies are basically ongoing. So my summary is, and for the discussion points, I think that interferon should not be forgotten, in particular if Bulevirtide is not available. Bulevirtide is approved by EMA. We see virological and biochemical responses in most patients. The issue how long to treat can be used in every decompensated patients. After having seen the New England Journal paper, now when to combine it with interferon, we may discuss during the discussion round. We have these additional new treatments which are in clinical development. And this is a teaser, this meeting here. If you want to discuss this in more detail, you need to come to Milan. Pietro may comment on this very nice meeting, the 3rd Delta Cure International Meeting, which will take place this year on October 11th to October 12th in Milan, again, hosted by Pietro Lambertico. And I thank you very much for your attention. Would be happy to discuss some of these points. And sorry for running fast through the slides, but Maura, there are a lot of data. Sorry.
Yes, you're right. Thank you for this overview, Heiner. I know it was a challenging talk.